Welcome to Click for IB Biology. Our topic is genetics, and in this particular presentation I'm going to be speaking about what's called non-disjunction in meiosis. This is a mistake, a fault mechanism within meiosis that leads to abnormal numbers of chromosomes in gametes and therefore in fertilizations. There's a blank version of this presentation, a PDF, down in the description. You can download that and annotate with your own notes. And then there's also down there, there's a 10-question quiz. You can answer those questions and check the answers to consolidate your understanding. So we're looking at the phenomena of non-disjunction, and then towards the end, we'll apply this within the context of Down syndrome in human beings. So there's actually two forms of non-disjunction, and the difference largely comes down to whether or not the mistake occurs in the first or the second division of meiosis. You'll recall that meiosis has 2 rounds of cell division. So let's just go through this. I'm illustrating this with reference to a homologous pair here. So this is our homologous pair. And we know that during the S phase, then the homologous pairs will replicate and the pairs of chromatids will be held together by the centromeres, and they will be aligned for separation during meiosis 1, so that when cell division occurs, what should happen is that one of the homologous pairs goes towards one pole and the other of the pair goes to the other pole, thereby separating the alleles and the genes. But in this non-disjunction phenomena, what happens is that the spindle pulls both of the homologous pair to one of the poles. And so I'm illustrating that at the next level, showing that after meiosis 1, we end up with both the homologous pair are in one cell, and then this cell here has been deprived of that homologous pair. Now, it's worth hesitating at this point to remember that, of course, in meiosis, we're not looking at the separation of one homologous pair, but all of the homologous pairs. So within a human context, there would be 23 such homologous pairs, like so, and they would be replicated like so and separated in meiosis 1 like this. And we would have to go through this for 23 pairs. So here we're only dealing with one of those to illustrate our diagram. But remember, there are 22, in human beings anyway, there are 22 other pairs operating in the background normally. So meiosis 2 then results in the separation of the chromatids. So these 2 pairs of chromatids here, they would align on the equator beside each other and separate their alleles such that this gamete cell here will end up with 1 extra chromosome, as indeed does this one, whereas these 2 cells here, they would lack a chromosome like this. So 50% of the gametes would be plus 1 chromosome, and then in the other side, 50% of the gametes would be minus 1 chromosome. So having an extra chromosome in the gamete or 1 chromosome less in the gamete, one could predict is going to have consequences in a fertilization. Let's take a look at non-disjunction 2. Now, non-disjunction 2, meiosis 1 continues normally. The fault occurs at meiosis 2, where we get, in this case, a failure to separate the chromatids. So this gamete here is normal, this one's normal, but this one here, both of the chromatids have gone to this pole in this direction, and for this pair anyway, none have gone to the other pole. The consequence of which is that this particular gamete here is plus 1 chromosome, and this one here is minus 1 chromosome. It appears from experimental observation and the frequency of chromosomal abnormalities that non-disjunction I is probably more common than non-disjunction II, and non-disjunction I would be particularly more common in females where the process of meiosis is arrested for a considerable period of time, allowing the joints between the connections between the chromosomes and the chromatids to become disrupted. Now let's apply this to an example, which is Down syndrome. In English, this is written as Down syndrome with the apostrophe. In the US, the S is missed off and it's known as Down syndrome. Named after John Langdon Down, 1866, the name was applied to Down syndrome was applied in 1866. He had previously discussed it in about 1862, I believe. And what we actually see here is what's called Trisomy 21. And so this is where we have 3 copies of chromosome 21. And this comes about because one of the gametes, so let's put an egg cell here. So for chromosome 21, this already has 2 chromosomes. And then if we have the sperm cell here, like so, it has 1 copy of chromosome 21, so that when they fertilize, you end up with this triple chromosome effect here. In this particular diagram, you can see that there's the presence of the Y chromosome, which is suggesting that this is a male. Okay, so trisomy 21 gives rise to Down syndrome. Just briefly on the topic of Down syndrome, because it's a syndrome, what this tells us is that there's a wide range of the characteristics in the phenotype in the individual who has this condition, and they can be from very, very mild to moderately affecting the person. There are characteristics of Down syndrome which are listed in medical textbooks and are not the subject of this video, nor is the additional outcomes of Down syndrome to be discussed here. So we know this is trisomy 21. Trisomy or monosomy, lacking a chromosome, can exist in a number of the other chromosome pairs, indeed in any of them, and it tends to have drastic effects on the development of the organism and their subsequent lifespan. Students who are interested in Down syndrome can research this in more detail. There's the link to the next video, the chance to subscribe to the site, and of course, our playlist of genetics videos. Thank you for listening.